Okay, so now I have the last talk of the session before the brainstorm session by Jeff 2. We'll talk about the clinical or precision oncology.
Great, thank you. All right, thanks to the organizers. Great to meet everyone. Yes, I'm James. I'm a faculty at Stanford. So excited to share with you some of the recent work we've been doing in thinking about how to use like these very large-scale, real-world clinical genomics data, both to design like in silico clinical trials and also to advance precision oncology. Maybe if I show the next slide, just like a quick question. So, how old do you think is the very first clinical trial? Anybody want to make a guess? 100 years old, 500? Yeah. How far back do you think clinical trial goes?
The Bible and Book of Daniel.
Okay. Yeah. Okay. So maybe you have seen this this before. Yeah. So actually, one of the first clinical trial, right, that I could find in records actually goes back to about 2,600 years ago, right?
My student.
Yeah, I've also given this talks before. So the idea here is actually a very interesting story, right? So the king of Babylon, the king of Nebuchadnezzar, right? So he had this idea, right, that if people eat more meat and drink more wine, that will make them healthier, right? So to test this idea, he had a decree, right, that all the citizens of Babylon, they can only drink wine and they can only eat meat. They can't eat vegetables and drink water, right? So of course, some citizens complained, right? And then to appease the citizens, he had to run like a comparison test, right? So over the period of about 10 days, there's one group of citizens who would follow the king's diet, wine and meat. There'll be another group that follow their normal diet of vegetables and water. And after 10 days, the king has people went around to measure the health status of these two groups of people. And much to his dismay, the vegetable eating folks actually looked healthier, right? So then he had to roll back some of the decree. Now, of course, over the last 2 millennia, right, clinical trials have changed quite a bit, but many of the core tenets stay the same, right? The idea is that we want to study, right, in a relatively well-controlled setting, the effects of a particular intervention on the health and outcomes of humans, right? So that's still the core idea. And in modern days, right, clinical trials is really the bottleneck and also the gatekeeper of essentially all of medicine. Right? So everything from COVID vaccines to cancer treatments, right? So you have to go through clinical trials. And the trials are also can be extremely expensive, right? Like each trial can cost on the orders of hundreds of millions of dollars. And most of the trials fail, right? So to account for the fact that many of these trials fail, the cost of actually taking one drug through all of these trials into patients is on the orders of several billions of dollars. So what I want to share with you today is some of the ideas we've been exploring about, okay, so given that these trials are so critical and they're so expensive, in parallel, we're also having a lot of these real-world clinical genomics data, right? By real-world data, I mean, like if you actually look at the medical records, right, of cancer patients, for example, you can look at other diseases, but of cancer patients, then we have, for example, databases like the Flatiron Network, which has like 2 million cancer patients in the US, right? For each of those patients, I know their first-line treatment, their second-line treatment, their biomarkers, and their outcomes. And in parallel, right, for many of these patients, we also have their mutation profiles because many of these patients have actually taken Foundation Medicine NGS-based tests, right, before they started first-line treatment. So, right, so from tens of thousands of these patients, we also know their tumor mutation profiles together with all of their treatment information EHR records, right? So those are the information that we could, in principle, link and derive from the medical records. So our idea here is that, okay, now that we have all of this data and say we can link them, right, can we actually use this data to sort of computationally run clinical trials in silico, right? And the benefit of doing this, I'll show you 2 applications. Is like, first, we want to see, can we actually use these computational or in silico clinical trials to actually help us to design the actual randomized clinical trials to make them more efficient and also more inclusive, more diverse. And second is, can we actually use these in silico trials to basically identify predictive biomarkers, right? So basically genomic biomarkers that can tell us, what treatments are likely to benefit which patients. Okay. So I'll tell you a bit about both of these applications. First, I just want to give you a bit of a context because people here may not be so familiar with the clinical trial. So first, I'll tell you about the first piece, right? Just how we can actually use these kind of real-world data, clinical genomics data, to help us to design clinical trials to make them more efficient and more, representative. And this is based on this work that's led by my PhD student, Rishan Liu, who's a fantastic PhD student at Stanford, that we published last year, where we developed this platform called Trial Pathfinder, which is like a computational platform that helped us to design these trials. I'll tell you a bit more about that in the next few slides. And we're very excited that actually this Trial Pathfinder platform is now actually used by pharma companies, for example, by Roche Genentech, to inform the designs of some of their new cancer trials. Okay, so what is the problem that we're trying to solve, right? So if you actually look at the design of a typical clinical trial, right, one of the biggest pain points, right, in designing these trials, one of the biggest complexities is to decide actually what patients to enroll in that clinical trial. That's often one of the biggest important decisions in a given trial. Right, so if you actually look at a typical trial protocol, right, so you'll have tens of pages of PDF that lay out specific rules that says that if you had this previous condition, or if your lab values fall outside some range, or if you are taking this treatment, then you're just not eligible to participate in our upcoming trial. So these people are recognizing that oftentimes the clinical trials are just overly restrictive, right? They're excluding too many patients. And this has huge consequences, both for the patients, because that makes it very difficult for them to enroll in trials. So in cancer, a large number of cancer patients actually would like to participate in cancer trials. That's to get access to cutting edge treatment. They're not eligible. It also hurts the drug developers a lot, right? So a substantial number of the trials just simply fail to reach the minimum enrollment requirement. And for every day it takes longer to enroll patients, actually it could be costing $1 million. The third consequence, which is actually very important, from a data science perspective, right? I guess it means that the fact that these clinical trials are often overly restrictive means that they are typically being evaluated on what are so-called Olympic athletes of the patients, right? And how well a treatment performs on these Olympic athletes do not really necessarily reflect how well it works on people like us, right? So there's a distribution shift, right? And that's people recognizing is really one of the huge weaknesses of the current design of clinical trials. Questions? Make sense? Okay. So people might be wondering, okay, so how do people currently design these eligibility criteria, and how do they end up being so restrictive? Right. So when they design the eligibility criteria for these trials, right, oftentimes, you want to balance the safety of the patients. You want to make sure that there's no adverse events. And also you want to sort of select the patients to highlight the efficacy of your treatment. Right? So those are sort of multiple objectives that people are trying to optimize for. And unfortunately, right now, there's actually very little or sort of relatively little evidence-based approaches for helping us to design these trials. Right? So most of that is based on sort of more anecdotes. So to illustrate this, I actually took a few screenshots of actual trial protocols being designed right now. Right, so some draft protocol, right? And the first thing that people do is that they'll just copy and paste over the trial that's done before by the same team, right? And then they'll go down like each line of these. So each line here corresponds to one of the rules that's used to exclude patients, right? So the first line says that, okay, if your neutrophil count is above outside of some range, you're not eligible to participate in this new cancer trial, right? And then you can see actually there's on the sidebar of the Google Doc, there'll be actually a discussion, or some would say, okay, can we relax this threshold from 1,500 to 1,000 to enroll more patients? Right. Somebody also pushed back and say, oh, we don't really have evidence to change it. And the third person then would say, oh, well, then just keep it to be the same as before, right? Because we don't know what to do otherwise. Right. And then for the next one, for hemoglobin, we have a similar discussion, right? Can you change it from 9 to 8 to enroll more patients? No, not enough evidence, right? So let's keep it to be the same as before. So the upshot of this is that, which rules are used to exclude patients very much depends on sort of the the history of the clinical team itself, or the history of the companies, right? It's sort of a very anecdotal process. And here I'm showing you sort of 10 different advanced non-small cell lung cancer trials, right? So each row is one of the trials, and each column corresponds to one of the commonly used rules that are often used by companies to exclude patients. Right? Some of these are based on biomarkers, lab tests, prior treatments, right? Or previous diagnosis. And I colored it to be yellow if that particular trial used that rule to exclude patients, otherwise colored gray. You can see that actually even across the same indication, often looking at the same kind of similar drugs or similar mechanisms, these are different immunotherapies, right? The different companies ends up having wildly different rules for building their cohorts, just because the process is so anecdotal for everyone. And everybody's recognizing this is a huge challenge, right? So for example, the National Cancer Institute said that the designs of all the clinical trials needs to be made simpler and more standardized. But how to do this in a data-driven way is challenging. And this is where we want to help with the trial Pathfinder algorithm. Okay. So that's the sort of setup for the problem, right? So now stepping back, as a computer scientist or data scientist, right? So then the problem at the core is really a data science problem about counterfactuals, right? So we have a set of patients, right, who are excluded from participating in these clinical trials. What I want to know is sort of the counterfactual of would these patients who are excluded, would they have, or which of them would have actually safely benefited from the treatment that's tested in the clinical trial? And the idea that we have for doing this is that it's to leverage the fact that many of the patients, even if they're not eligible to participate in the clinical trial, after the drug is approved and after it comes to the market, many of the same patients ends up taking the same drugs after it comes to the market. Right? So many of the drugs are actually even used off-label after they come on the market. Right? So then if we could just identify these patients in the medical records, right, then we can try to answer these kind of counterfactuals, right, in a systematic way. And to do this, we leverage the data from the Flatiron network that I mentioned before. We have EHR data from a large number of cancer patients. They cover about 2 million cancer patients in the US across many 200 plus different clinics in the US. And for many of those patients, right, so for all of them, we have sort of the treatments and vitals and biomarkers. And for many of them, we also have the genomic profiles. Okay, so here's sort of the schematic, right, of how we do this. I want to give you an intuition, and the details are in this paper. So the idea here is, here's how we actually use the EHR to actually computationally emulate the trial in silico. Right, so say I have a particular trial. The trial will have some treatment drug, right? Maybe an immunotherapy, will have some control, could be a chemotherapy, and we'll have a set of rules for excluding patients. In this case, maybe you only want to have patients with bilirubin less than 1. So bilirubin is a commonly used assessment of liver function. So then what we do is that we go into our medical records, right? So we can generate synthetic cohorts. We'll generate one cohort of patients who actually took the treatment, right, across all these different clinics, and who satisfies this threshold, bilirubin less than 1. We'll generate another synthetic cohort of patients who took the controls, right, also satisfy the threshold. Now these two cohorts of patients will look very different. Maybe they have different demographics and different distributions. So then we can sort of computationally match these distributions to make them comparable, right? Just as an aside, right? So to do this matching, there's a lot of interesting, this is where there are a lot of interesting machine learning statistical techniques, right? So you can do things like you can use propensity-based methods. Since the data is very high dimensional, you want to use some sort of machine learning predictors on top of the propensity-based methods. We've also developed approaches based on sort of optimal transport, basically also another approach for aligning these data distributions. But the main point here, and I can describe this more in detail, but the main point here is that you want to basically match these distributions so they're comparable to emulate sort of this randomization process in the actual clinical trial. And after you do this, then you can sort of measure things like what is the hazard ratio and what is the outcome of these two synthetic cohorts. And because it's all done in silico, it's actually very easy for us to computationally change these different thresholds. Right? So I could say maybe bilirubin less than 1 is too restrictive. What if I change that to be more inclusive, to be less than 2, to enroll more patients? Right? So then I have larger synthetic cohorts because I can find more people in the medical records, right, who satisfies the broader rules. but then I can do the distribution matching again and look at the outcomes. And the idea here is that by computationally changing these thresholds, if I don't actually see any difference in the outcomes, right? If I don't see more adverse events, if I don't see worsening of the survival of patients, then that gives us evidence that perhaps we can actually broadening this restriction of the bilirubin values, right, from 1 to 2 or within this threshold. So that's the high level intuition. So at this point, you might have maybe a few questions, right? First question you might ask is, okay, so what if the drug is actually not on the market? Right? Because everything I've said so far relies on the fact that we have to look in the medical records to find patients who are taking these drugs. Right? What if it's not on the market? It's a good question. So oftentimes what we do here is we leverage the fact that you actually often have drugs of similar mechanisms of action, or even the same drug that have previously been tested before that are on the market. Because actually, most of the clinical trials, especially in cancer, obviously are testing a run on drugs that are already approved for some other indications. So we have the information from those that are found in EHR. The other thing you might wonder about is that, okay, so what about confounders in these observational data, right? So here we're looking at non-randomized observational data. So that is a true limitation of any this kind of analysis. We do benefit here because we have very highly curated EHR data, right? So we follow patients over many years. We have their genomic profiles and their treatment and outcomes. But in the end, we also validate our results with data from actual randomized clinical trials. Yeah.
Direction, but the way that you're sampling the drugs is very highly biased, right? Because I mean, yes, drugs may not be on the market, but really these clinical trials are trying safety and get weeded out early on.
Yeah.
And so the population of drugs that survive may have very different risk behavior in the overall population because they have been vetted through real clinical trials. Isn't that a problem in the long run? I mean, similar drugs and same mode of action set aside, but something that is really novel.
Yeah, so it's a good question. So what we try to do is we want to be representative of drugs that are in sort of phase 2, phase 3 stages of clinical trials. We actually go to the clinical.gov and basically curate all the phase 2, phase 3 drugs. Right. So it's true that even the phase 2 drugs are certainly a selected set of all the drugs, right? But that's from the ones that ends up making it the most far and have the biggest impact.
So you were only measuring the hazard ratio there. How do you take care of the adverse events?
Yeah, so we have multiple outcomes, right? So we have mortality. This is one outcome. We can look at time to progression, right? Time to death. We can also have measures like time to adverse events. Right? Adverse events here could be some toxicity events.
How do you weigh all of these things across each other?
So right now we don't weigh them. We just actually look at each of them separately, right? So because we can just see, okay, how does changing this eligibility rules affect adverse events, affects survival, affects each of the outcomes.
Still a subjective assessment after that, whether we want to.
So we'll actually end up seeing that there is a sweet spot where you can actually relax these rules and actually get sort of a Pareto optimal for all of these for all of these metrics. Yeah. Okay, good questions. So just to summarize, right? So here's the approach, right? So we start off with this trial protocol, which is written in natural language. We can do some NLP to basically extract computational rules, right, of defining the cohorts from these trial protocols. Those are encoded as logic statements. Then we actually go into the actual medical records, right, of these millions of patients to generate our synthetic cohorts, like I described before. We can sort of computationally match them, right, using the statistical approaches, and then look at the outcomes, right, by all of these metrics, right, the adverse events, survival, progression, et cetera. And we don't just do this once, right, because this is all in silico. We actually do this millions of times in parallel. Each time we do this, we actually use a different combination of eligibility rules. And the reason why we do this is to try to really assess the impact of different specific eligibility rules, because there could be interactions between them. And at the end, right, after these millions of trials, then we sort of summarize all of these results together using this sort of technique that we developed called sort of criteria Shapley values to summarize the impact of each of these eligibility rules. So just to give you a sort of concrete sense of what this looks like, right? Again, going back to our example of the bilirubin, right? So here are these 10 different advanced non-small cell lung cancer trials, and they end up using different bilirubin thresholds for excluding patients, right? So for each of these trials, which is one of these curve here, we can actually then have in silico trials, right? That says like what this trial would have looked like if I just changed the bilirubin threshold, but holding everything else fixed. Right, so each of these curves here corresponds to the counterfactuals of changing the bilirubin threshold, and the y-axis is basically like a hazard ratio. Right, so lower is better. But the main point here is that actually within a reasonable range, right, you could actually change the exclusion criteria of this rule by making it more inclusive, right? Enroll more patients without actually hurting yourself in terms of the hazard ratios, right? Without engendering safety of the patients. And then by doing this systematically, we come up with sort of a set of data-driven recommendations, sort of guidelines for these lung cancer trials. So we applied this to a bunch of different cancer types, like breast cancer, melanoma, but here I'm showing you the results from the advanced non-small cell lung cancer. So we applied it to sort of 10 different trials from different pharma companies. And maybe the most important thing is sort of the last row here, which is sort of the, what these trials look like under the original designs from the pharma companies, right, that's proposed, and what it looks like under our data-driven criteria. So on average, we show that by taking this data-driven approach for design and eligibility of these trials, we can more than double the number of eligible patients, while actually still reducing the hazard ratio overall. Right, so this means that actually many of the patients who were systematically excluded from these trials before, they would have actually benefited to a similar or even greater extent compared to the more Olympic athletes of the patients that were eligible. And importantly, the data-driven approach we have here actually enables a lot more women and more minorities and older patients to access this trials. Many of them were systematically excluded before. So just to summarize this part, right, I think this is sort of one first step to taking a data-driven approach to design clinical trials to make them more efficient and safer and more inclusive. I didn't have time to talk about here, but we actually validated our designs using data from actually randomized clinical trials, right? Because there's actually turned out to be a lot of natural variation, which gives us an opportunity to do sort of natural experiments. Also using data from different cohorts outside of Flatiron. Here we mostly talk about cancer, but I think similar kind of methodologies can also be extended to other disease types. And we're very excited that we published this paper last year, and this actually now is being used by pharma companies. It was also recognized by the pharma industry as one of the top data science innovations in the past year. Okay, so more briefly, so the second part I want to mention is how we can actually use these similar kinds of approaches in silico trials to do precision oncology. So this is based on a paper that we wrote, which just came out last week. So the idea here is that I mentioned that for a large number of these patients, right, in addition to their medical records, we also have their genomic profiles because they've taken the foundation medicine testing, right? And we have these profiles before the first line they start treatment. And the data we have is usually about, you have NGS sequencing for many of the several hundred of the key cancer driver genes. And what we're very interested in is to look for, yes?
Just to make sure, this foundation data, the genome-wide or the panel data, is something only like 5 mutations per individual. So is it enough data to go?
Yeah, so they basically did search for a panel made for about 600 or 500 plus genes, and then they do quite detailed, like they do NGS sequencing for each of those genes. And we actually do find, so this is actually what's shown here, we actually do find quite a large number of mutations for many of the patients. And we look at this across 8 of the common cancer types. And what we are really interested in doing from this is to basically look for mutation treatment interactions, right? By taking this synthetic digital cohort approach. So what does that mean? Like, what do I mean by mutation treatment interactions? Right. So for example, this is one kind of interaction we identified, right? So if you look at all the bladder cancer patients, right, we'll go to our medical records and find all the bladder cancer patients. We can generate a cohort of all these bladder cancer patients who are taking immunotherapy, right? So there's a large number of them. And then we can find, okay, so among those bladder cancer patients who are taking immunotherapy, some of them will have a mutation in this gene called APC, right, which is a tumor suppressor gene. And these patients actually end up having much better survival outcome compared to other bladder cancer patients who are also taking immunotherapy, but did not have mutations in this gene APC. Right? And conversely, we can also ask the complementary question, right? Among all the patients with APC mutation, now is immunotherapy actually the best treatment for them? In this case, it seems like the ones that are taking immunotherapy also have better survival outcomes compared to the ones who are given other treatments, could be chemo or targeted therapies. Right? And mathematically, this corresponds to basically looking for these kind of interaction effects, right, between the mutation, which is the genotype here, and the specific treatment, which in this case is the immunotherapy. And because we have these large cohorts, we can basically do this systematically for every single mutation and for every treatment that's on the market. Basically look for all the possible interactions. Yes.
So the data that's part of the foundation, it's not a randomized trial, right? It's basically patients who asked for the foundation panel to be done on their, isn't there already a bias in there? The type of patients who go for that one? Are you missing something here?
Yes, so it's a selected set of patients, but it's, the test is fairly commonly done. So there's like 200,000 patients who've taken the foundation medicine test. I don't think that bias necessarily affect this kind of results, right? Because this says that, condition on taking a foundation medicine test, right? There's still a very strong interaction effect between whether you have APC mutation versus when you don't have APC mutation. But that's a good question because the next thing we did is we validated these findings using data from actual randomized clinical trials. Right, so the previous results all come from looking at the medical records, and we look for these interactions. But in this particular case, right, we actually, so Roche actually ran a randomized clinical trial of bladder cancer patients when they're given Atezo, which is immunotherapy. Right, they also done foundation medicine tests for all of these patients in the randomized trial. And we can identify sort of similar kinds of effects, right? That there is this positive interaction between APC and immunotherapy. So patients who had APC had much better survival outcomes when given immunotherapy, exactly as we had predicted from our initial analysis.
Question?
Yeah.
When you're talking about a real situation and not in a clinical trial, there is often a situation that according to the response of the patient, the drug is changed, the regime is changed. So you don't typically have the whole survival curve because events happen along the way.
Yeah. Yeah, it's a good question. So there we look at multiple outcomes. So overall survival is one common outcome. We can also look at time to progression. So what that means is that how long did this particular immunotherapy work before it stopped working, so the patient had to move on to the next line of treatment. So that time itself is actually a useful indication of how effective that treatment is. So we also similarly show that that similar kinds of APC immunotherapy interaction, even when we look at time to progression to the next slide. Okay, so that's just one example. But the power of doing this in silico approach across all the possible mutation treatments combinations that we can actually now discover from the real clinical data, right? A much larger number of these kind of predictive biomarkers. Here I'm just showing a subset of them, but in our paper we actually have a full list of over 400 of these markers. So each marker will be similar to the APC story, right? So for example, here you look at a particular kind of treatment, like maybe immunotherapy in advanced non-small cell lung cancer. Right, and then for a particular treatment class, we can see that if you have KRAS mutations, then you're more likely to benefit from immunotherapy. If you have EGFR mutations, you're less likely to benefit. Right, so the color here, if it's blue, that means that there's a positive interaction between that mutation and treatment. Red means that there's a negative interaction. And we're very excited about this as a potential, first to generate a lot of interesting research hypotheses, but also as a way really for treatment recommendations for precision medicine. Right? Because each of these predictive biomarkers leads to a very specific and actionable treatment recommendation of which treatment is likely to benefit that particular patient.
So going back to Rodette's first question, you were showing it on the gene level, but are you clustering together all the mutations in the gene, or is it typically one mutation?
Yeah, so we look at different resolutions. So here, just for simplicity of visualizations shown at gene level, but we also look at very specific subtypes of mutations, like for example, like translocation, truncation. So we'll have that subtypes. And also, we also look for like sort of gene-gene interactions, right? Basically, we have enough data to show that, maybe it goes back to one of the earlier questions, we have enough data to show that the same patient actually has multiple mutations, right? Maybe they have both like ALK translocation, but in addition to that, they also have a mutation in TP53 or STK11, right? So then we can look for that interaction effect of how much that patient, right, would benefit from an ALK targeted therapy if they have an additional mutation in TP53 or STK11. Right, so here are basically the kinds of interaction maps that we can identify here. For example, that says that if you have a patient who are given KRAS targeted therapy, right, and if they have additional mutations in APC, then that actually means that the KRAS therapy itself is going to be much more effective. Right, so that's the blue here. I think that there's a positive interaction. So we together with the paper, we also released or created this precisioncancer.org, which is to visualize and summarize all of these results we have here. So there's a lot of data and information here of the specific mutation subtypes and interactions with all of these treatments. And so hopefully this will be like a useful resource for the broader research community. So just to summarize, I think the core technical idea here is that now that we have these linked large clinical genomics data, right? I think one really promising way to leverage that data is to basically use it to run in silico trials. And we've shown here that you can actually use these in silico trials to help us in designs of the actual clinical trials to make them more efficient, more safer, and more inclusive. Right. So that's really one of the key mandates of the NCI and of the FDA. And we can also use them to discover these predictive biomarkers, which can be greatly helpful for precision medicine, precision oncology. And there are certain limitations of working with this real-world clinical oncology data, which many of people here have mentioned. These are real limitations, which is why we do still think that it's very important to validate all of our results with the actual randomized clinical trials. So I'll stop here and just want to acknowledge again my student Rishon, our collaborators at Roche and Genentech, as well as other collaborators at Stanford for all of these projects.
Question regarding the first paper, the Nature paper. So you're basically using this to optimize cohorts for future studies. You also mentioned that correctly that the previous trials were often done on unrealistic cohorts, which are not really representative of the real population. So I think you could also use this framework to reassess the existing drugs on the market by essentially in silico reconstructing the criteria of the original study and then comparing the performance of the drug on the subset of patients to the whole population of patients in order to see how much worse, different, better the drug actually performs on this, extended cohort.
Yeah, that's a great question. We had looked into that, and it turns out that there is a systematic overestimation of the results in the clinical trial compared to the results in the real world we see here, but that also has been reported previously in many other studies. So to people, there's a growing recognition that the clinical trial results, even if it's randomized and perfectly controlled, right, since being biased upward in terms of their effect size.
So after a drug has been approved, are there additional data for future, like people who take the treatment, do they do better, do they do worse? I mean, that would be more realistic.
Exactly. So that's exactly the data that we use from the EHR, right? Because we know that the drug's been approved, right? This person on the medical record, right? They take that drug, and we know their outcomes. Exactly. Right? So that's exactly the information that we use. Yeah. So that's why we go to these hundreds of clinics, right? That create that flat iron to look at millions of cancer patients who actually ends up taking these drugs after they've been approved. Yeah.
Okay, thank you.